Hello, everybody. My name is Siavash Raisi, PhD student from UCSD. We developed a new tool for aligning the optical map data and the SV detection pipeline named FaNDOM. FaNDOM, or Fast Nested Distance Seeding of Optical Maps, is an optical map alignment tool and SV detection pipeline. FaNDOM works based on the novel seeding algorithm to rapidly reduce the alignment search space. As you know, optical map data are high coverage, more than 100 or 300 of the genome, and aligning 2 million molecules would take more than 700,000 CPU hours. So having a very fast and robust alignment is very necessary. Our tool is an independent tool that works mostly for the Bionano Saphyr data, and on average, it takes 1.5 CPU seconds per fragment, and it takes 1 hour for the sample for each sample to run. Um, there are two main. Our tool is faster than the existing tool like the Ref Aligner or the Blast. Um, as the results show here, as we can see, the FaNDOM is 4 to 6 times faster than the Blast and 13 to 14 times faster than the Ref Aligner. Uh, Also, for calculating the precision and the recall, we selected 207,000 molecules from each cell line. This cell line, Na12878, GM09888, and so on, as you can see their name here. Um, but it is not trivial to designate an alignment as a correct or incorrect. So for detecting the true alignment, we align all these molecules with these three tools. And then we apply the majority rule. If an alignment at least reported by two tools, we assume it as a true. As we can see here, the precision and recall of the FaNDOM is much better than the Blast, and it is really close to the Ref Aligner, but it is 13 to 14 times faster than that. So it is worth to use the FaNDOM as an aligner and SV detection pipeline. Also, FaNDOM has a very sensitive detection of the partial alignments that enables our SV detection pipeline to have a discovery in cancer cell line or other cell line for detecting the structural variation. Um, in the COVID project, we use the FaNDOM as an independent tool to kind of confirm all the finding by other tool, by the Bionano pipeline. As you can see here, this is the result of the FaNDOM. This table is the table 2, which is the list of the unique structural variants overlapping with coding regions of the gene, which are identified in the patients with the COVID-19. As we can see here, for example, lots of unique structural variation that are detected by by other tools like the Bionano pipeline are also confirmed by the FaNDOM. There are only 2 of them that were not detectable by FaNDOM because the resolution of the finding, the inversion or deletion inversion less than 30 kilobase were not detectable with FaNDOM. So more than 90% of the structural variation that were found by the Bionano pipeline are also confirmed and found by the FaNDOM. Here is the table that all these green ones are detected by the FaNDOM. And this is the continue of that number. All these 16, except 2 of them, are detected by the FaNDOM. In the continue, I'm visualizing 2 of our findings and show how we detect them. Here is the example. This is the duplication that find in the chromosome 1 in the gene, in these 2 genes. As we can see here, also this visualization made by map optics. As we can see, there is a one purple region here that is duplicated. There is 2 partial alignment for the context to the molecules. As we can see here from the label, 1350 to 1382 aligned to the purple region. Also, the label from 1386 to 1419 again aligned to the purple region. I have some schematic like that here to explain what is going to happen. Um, as we can see here, this region, the purple region, is got duplicated in the query, which is the signature of the duplication in our case. And this is the screenshot of the uh, mm, genome. As we can see, this region of the duplication contains these two genes that exactly we wrote their name here, H heter1 and edart. This is the example of the this is the example of the another structure variation that were found by FaNDOM. This is the duplication in the chromosome 19, the gene ZNF443. Again, there is two purple region that align into different region in the query, in the context that we had. I have again the same schematic for that. As we can see here, these two green regions align to this purple region on the reference. So this is the signature of the duplication, and we find the duplication in this gene, in this chromosome. Here is the screenshot of the genome browser, and as we can see again, this gene is got duplicated. And thank you so much for listening to me.